Welcome to today's seminar. Our speaker today is Conchita Alonso, and Conchita is a plant evolutionary ecologist, NPI here at the EBD. And today she's going to tell us something about her work on plant epigenetics. Go
And yes, today I will speak to you about a topic that has interested me for the last 15 years, that is plant epigenetics, and how this could help plants to survive. And let me first introduce our team. That all this work I will be talking today started, as I said, like 10 years or 12 years ago, and it has been conducted together with Mónica Medrano and Carlos Herrera, and with the skillful assistance of molecular and chemical experts. Pilar is the only one still with us, and also Javier Valverde, that was postdoc in our lab, contributed to the last part of the analysis that I will show today. And for the experimental part, many students along the years have been helping us, and they are agreed here. Okay? So this is the outline of the talk. I will first give you some introduction with about the specific features of plant epigenetic regulation, and then I will speak about an experimental approach to understand epigenetic variation in response to stress, and then something more conducted in nature to aiming to understand the difference between genetic and epigenetic variation in wild plant populations, and then a little bit of wrap up with the conclusions that we can learn from there. So what is epigenetic regulation? So for ecologists, we train, I have made this graph to explain people from other fields that phenotype for us is the interaction between the genetics or the genome and the environment. And in this framework, we can find epigenetic variation as an extra layer of variation between genotype and phenotype. And here you can find that how we can study epigenetic variation, and we can do it like with markers that are specific, or like an average global methylation variation. And the epigenetic mechanisms are all related to each other. I will be analyzing, or we have only studied DNA methylation, but DNA methylations is related with histone modifications and the small RNAs, and all of them contribute to transfer the information that are in the genome and contribute to phenotypic variation. And I would like to highlight some of the features of epigenetic variation differences compared to genetic variation. Some of them, at least, can be triggered by the environment, and it means that several individuals in a population can reach the same variability because the environment is shared. They are transmissible, but also reversible. The rate of change is several orders of magnitude higher than for the genetic variability. And the transmissibility could be transgeneration or for several generations. And then it's highly variable. And I will be saying something else about these features now. So in plants in particular, in other organisms, most of the methylation is in cytosines that are in the sequence followed by a guanine. In plants, we have also methylation in all the other contexts, and this is thanks to a specific group of methyl transferase that are the chromomethylase that are with this acronym. And in this study, they have analyzed the genes for these enzymes in more than 400 species, and here is the proportion of species that have different chromomethylase, and you can find, for instance, that chromomethylase 3 and 1 are specific of eudicots. So there is a long evolutionary record and relevance of methylation in plants. Also, this methylation in CHG and CHH can be found in any genomic location, not only in the promoters, that is the main place where you can find it in animals or other organisms. We have a lot of methylation related to within genes, in the body of the genes, and also associated to transposable elements. And that means that there is a broad variability in the global methylation level that genomes of different plant species can have. Here is, you can see that from Arabidopsis, that is here in the corner with 4%, and to some of the other species that reach 40% of their cytosines are methylated in the genomes. Animals, we are more or less in the scale of Arabidopsis. And so only some insects have something higher. So methylation seems to be very important in plants. I don't know exactly how much in other kind of organisms. That's why we became interested in epigenetic mechanisms. And also it's very dynamic, as I said before, and it's dynamic between tissues, between ages, and also very interesting, that's something that we have also found, is that different modules of an adult plant can have different methylation markers and also different methylation levels, and this is transferred to the seeds and progenies that you can obtain from different modules. And that's like another scale that plants have, and that is distinct from animals. Very relevant for ecologists also, DNA cytosine methylation change in response to stress. And here I have shown two old reviews. The first one suggests that methylation, epigenetic changes could be related to the priming effect. So why individuals that have already experienced stress can better to a subsequent stress. That's priming, and they were pioneers in suggesting that this could be an interesting phenomenon to study when you analyze stress response in plants. And also a literature review we conducted several years ago, with which we found 49 studies that have checked the changes in this market according to different stresses. And what I wanted to highlight here is that the results of these experiments was very variable for any of the stress factors that you can. So in some studies they found no changes, in some ones equal gains and losses of methylation, and in others there was a net increase or a net decrease for the same stress, depending on the experimental procedure, the species, and so on. And we will pay attention for the rest of the talk to draft stress that is included here. So you can see more studies address this, and very variable across species or experimentals or studies. Okay, so we move from the introduction to the first part of the experimental results that I will show you. This is my favorite study area, Cazorla Mountains in Jaén province. It's a mountainous area, as I said, with a heterogeneous landscape, particularly rich in plant endemics, in endemic plant species. And here I saw, well, more or less you know where we are, but the precipitation is very variable along the season and across years. And that's not, it's a characteristic of the Mediterranean environment. And I said that the communities in this area are on risk because we are aware that climate change is increasing temperature in the last decade. That is here in this, here's highlight. We have already increased more than 1 degree in the surface temperature change on average. And we are almost reaching this 1.5. And in any case, with any of the predictions or scenarios for just 1.5 or even 4 degrees, something that is very clear for Mediterranean is that the time under severe and strict drought is going to increase. If we reach here, quite a lot. If we can keep here, still quite a lot in the southern part of Spain. So we are interested in drought. The plants are experiencing longer periods of drought than they were expecting. So we took one model species to work in the greenhouse. Rhodium cicutarium, that is a generation, is an annual heir, and that's easy for the experimental designs. It's a spring flower native to Mediterranean, but has become invasive in many other places in the world. We collect seeds in 2 places in more or less a flat area, but high, and this is more rocky, Puerto del Tejo. This was the population I have in my first view. So more or less you can see you can have in your mind the scheme and where are the plants. This is the experimental design, and we took seeds from plants that were in the field. We grow them in the greenhouse for a generation to homogenize the environment in which they grow. And today I will be talking to you about the F2 that follow a factorial design in which we apply a chemical to demethylate the plants so that we erase somehow the memories, and then water stress that was applied like a recurring water stress, meaning that control plants were watered every week, and the others, the treated plants, were watered every 2 weeks. So they were experienced along the whole life after the establishment period, okay? And this is what we measure. We measure global DNA methylation in leaves and roots at seedling and adult stage, and then several phenotypic traits. And today I will pay attention to phenology, the time to flower, and reproductive effort that was measured in different ways, but today I will just talk about the total number of inflorescences. And the data were analyzed with linear mixed models, including the family as a random factor. Okay, our hypotheses were that changes in DNA methylation that we induce experimentally will have a consequence in phenotypic traits that are relevant for fitness, the two traits that I have mentioned. DRAF will modify DNA methylation and also have an effect in these plant phenotypic traits. And for some of the traits, we expected an interaction to occur, meaning that if we modify DNA methylation, the outcome of the treatment will be different. Okay, here are the results. So seed demethylation that is indicated with different colors, plants treated with azacitidine delay the flowering in the 2 populations. There is also a significant population effect, meaning that this, well, in particular this, the flower later, and also there was a interaction with DRAF that was seen that the DRAF delay flowering only in CH plants and only if they were not treated with the azacitidine. And here is like a marginal 3-way interaction that is explained by this. Okay, so we found, well, for phenology, that's what we found. For the reproductive effort, both the methylation and DRAF reduce the total number of inflorescences. The total number of inflorescences is a good estimate of fitness because this species is able to self-pollinate. And in the greenhouse, most of the fruits produce most of the flowers produce fruits, and all of them have 5 seeds. So it's a good estimate. And as you see, there was a scan effect of azacitidine, of water, and again, we have an effect of population, meaning that plants in this population produce less inflorescence in common garden conditions in the greenhouse. And as regards the effect of the treatments in DNA methylation is, as I said, we measure methylation in leaves and methylation in roots. In control plants, we can see that roots tend to have a higher methylation level. The equality will be along this. And also we found a significant positive correlation, meaning that plants that have a high methylation in leaves also have high methylation in roots, and the opposite. But when we put in those plants that experience DRAF, DNA methylation and roots was not correlated, meaning that the balance of the methylation level between tissues above and below ground was somehow altered by the stress. And this is something that is quite interesting and that was not previously accomplished, shown by other people. Well, this study is under review, it's in the archive, but hopefully soon will be published in somewhere. We also wanted to have a deeper molecular understanding, and then, and to search for some collaboration, and Francisco Balao in the University of Sevilla and Ovidio Pam were our collaborators for doing this. We have to build a reference genome, a draft reference genome for the species, and we apply the methodology to a subset of our design, meaning that out of 288 plants, we can just have money to do this in 32 individuals that were from the 2 populations, 4 families, the 2 demethylation levels and the 2 water levels, and just 1 individual per family on each of the treatments. So we don't have replicates per family. And the protocol that we follow was developed at a lab and used an enzyme digestion to reduce the target loss in the genome, so to have something more skillful. And then you have to do bisulfite conversion, prepare the libraries that Pilar developed here, and then the bioinformatics analysis that we have help from Frank to accomplish this. And here are the results. So in leaves, with this, we follow this protocol just in leaf DNA. The root DNA is still waiting for our analysis, but soon will come. So draft and azacitidine exhibit a significant interaction, and meaning that the individuals that suffer draft in yellow here, oh my goodness, and were treated with azacitidine at seed state, increased methylation. So this was something that we didn't expect, because the methylation reduces methylation, and draft as a whole also reduces it. But when you analyze with this method, we found a significant interaction. Here we can see that in well-watered plants, the azacitidine reduces methylation, and this in the CG context. And this was the expected, because azacitidine blocks one of the methylase enzymes, the methylation enzymes that affects mainly this context. But it's interesting to see that in the other 2 contexts, there are changes that have gains and losses of methylation at the same proportion, and that's also interesting. And still, we also have many loci in which the methylation that was in the control plants is lost. That's also interesting. So it's a net decrease, but also increases in methylation are found. And the most striking part is that there are more than 4,000 loci in which there is an interaction between the 2 treatments that we will see better with this representation, in which the 4 groups of plants are represented, and we found altogether more than 6,000 loci to analyze. Most of them were in the CHH context, some of them also in CG, and the context with less number of significant changes was CHG. And most CG changes are in exons, in gene. Well, the gray part is always big, but as we are working with a draft, many of those places are places that we don't know exactly what they are. So I will not pay attention to the gray area, pay attention to the color area, and then CG is mainly in exons, and whereas CHH is mainly in transposable elements, and very little ones in exons, so it's introns and transposable elements, where these are located, remembering that these are the main changes that we find. And for those that interact between the 2 treatments, we can find up to 8 groups, and then the representation is simplified by these 8 groups. In the first cluster, we have 4 groups of changes that mainly are here, these 4, that the changes are they are mainly non-methylated loci, whereas in the other group, most of them were more frequently fully methylated, like in red here. And then there are these two clusters here that show transgressive effect, meaning that control plants and double-treated plants were either non-methylated or methylated. And this is an interesting pattern that was not described before either. And we could not do annotation of all the loci. It's not feasible. They are 6,000. But we found 29 regions in which, in a short distance, you can find more than 10 loci that change methylation. And those were annotated, and you can find all the who are they in the paper. But some of them are related with methylation, abiotic stress, or transcription. So they are functionally related with the activities that we were expecting. So just to have conclusions, okay. From this first part, we will say that DNA methylation and DRAF have an impact on fitness. Seed demethylation delay flowering and reduce reproductive effort. DRAF reduce reproductive effort and alters the balance of methylation within the plant. The expected interaction is hard to find at phenotypic level, mainly because the two factors have detrimental effects, and also because there was a variability between the two provenances that we obtained. And this was also intriguing and related with the next that I will do. And then in leaves, we found that there is a strong interaction at molecular level. So phenotypic level was more difficult to find because there is variability across individuals and across provenance, but at the molecular level, the interaction occurred, meaning that DNA methylation is related with the response to DRAF stress. Okay, the second part, as we found these differences between the two provenance, we were intrigued by that, and it could be genetic or epigenetic. And how to decouple genetic variability from epigenetic variability in nature? So one possibility is to investigate the relationship between genetic variability and distance between individuals. Is the framework of the isolation by distance framework, in which here, if you pay attention to the blues, we can have no null model if there is no relationship between the relatedness of the individuals in natural populations and the distance between them in the population. But very frequently, neighbors are more genetically related, and the relationship degree decreases with the distance. And if that is the case, we have this kind of linear model in which distance in the space is in the x-axis and the similarity between individuals in the y-axis. And if we have this null model, then we can continue comparing with what will happen the same with epigenetic markers. And I will go through this in the results, okay? But if we have genetic variability, spatial structure, and this linear model, we have a null model to check if they are similar or not. Okay. So here are the individuals that we sample in the field. This is one of the populations. This is the soil. It's a flat and continuous green, deep soil, whereas the other is more rocky, more steep, and well, you see that this is the distance between pairs of individuals, the distribution, and you can find that there is a continuous distribution, whereas in this, the distance between pairs has this kind of multimodal distribution, meaning that they are grouped. Well, you can see here also. Then we divide this distribution in 6 categories of distance according to the percentiles, and we analyze the genetic relationship and the epigenetic relationship based on markers that are anonymous, AFLPs for genetics, MCPs for epigenetics. And what we found is that in the first of the populations, the neighbors here, these are the neighbors, were more genetic related than expected by chance. So our first prediction that the neighbors are somehow more family related than the others is fulfilling this. We also found that for epigenetics also is the dot is, and that is significantly higher than expected by chance. And then the relationship decreases with distance, and here is the model. So we have a null model, meaning that the genetic relationship decreases with distance, and for epigenetics is lower. In the second population, we found more or less the same genetic pattern, but for the epigenetic relationship, we didn't find any significant relationship with the distance in plants. If we have two populations, I'll be able to know what is most common. But indeed, we did 3 populations that we didn't study in the greenhouse, but we have collected data in the field. And the third population, again, is more flat, not as deeper soil as this one. The individuals were grouped, and despite being grouped, we could find a linear relationship and negative relationship, both for genetic and epigenetic relationship. Meaning that for the species, we can say that plant neighbors are more genetically similar than expected by chance. The genetic variation is more spatially structured, meaning that the slope of this relation is higher. And only in this population, in Puerto del Tejo, epigenetic variation is not spatially structured. So according to our several possibilities out of this theoretical framework, here, Erodium secutarium, in most cases, will be in this scenario in which epigenetic variation is unresponsive to local environment, meaning that the slope is lower than the genetic variability. We can also summarize this study by doing a path analysis in which, in addition to explore the relationship between spatial distance and genetic distance, and here between spatial distance and epigenetic distance, we can explore the direct relationship between the 2 factors, okay? Meaning how much we can explain from the epigenetic relationship from genetic relationship. And we did this applying it to interspecific comparison, because indeed we were interested in knowing how this endemic plant species that is very rare, is called Erodium cazorlanum because it's mainly distributed within the Cazorla mountains, is able to use epigenetics to survive in this environment, because it's not widespread as the other. No? So our question here was to compare the patterns in the space for 2 species that are related in the same genus, with similar dispersal agents, but with contrasting distribution range and a habitat specialization. And we did the same study in 3 populations. In this particular case, all the populations where the individuals were grouped, meaning that there are these kind of distributions that are not continuous. And in all cases, what we found was reduces similarity among neighbors. So for neighbor plants, in some cases, the relationship, genetic relationship was random, not different from the random expectation. And in some cases, there was no linear relationship with space. And the genetic spatial structure was less steep than in the previous species. And in none of the populations we could find a spatial structure of epigenetic variability. We conduct this study not only in Erodium, we did pairs of species in 6 different genera, some of them widespread, some of them restricted to mainly to dolomites, but not always. We altogether analyzed 42 populations and more than 1,000 individuals with these anonymous markers, and we select the species to have similar dispersal agents but contrasting distribution range and habitat specialization. And altogether the model show up that epigenetic, so the relationship between spatial distance and genetic distance is more or less similar, but the main difference appear here in the relationship between genetic variability and epigenetic variability, showing that this is more or less clear in widespread species, but in those that are restricted to specific habitats that are much more stressful, is not so much. And what we can, how we can interpret this? We can interpret this in different ways. One is that microhabitat might be very important, and maybe the limiting factor is not changing linearly. And this means that the epigenetic relationship or the epigenetic variability is locally determined, and also that the changes between generations, the transmission between generations is also lower. So the take-home message from all these studies will be from the experimental approach that epigenetic response to stress could vary between provenance, is not identical for all the individuals in one species. Most of the studies usually generalize the experiments for the whole distribution range of the plant and doesn't take this into account. Stress can decouple methylation above and below ground, and this is also something new that we usually in the field we sample leaves because it's easy, but maybe the main responses are in roots, and in that roots were the organ or the tissue that was changing more in response to stress in our experiment. But this is really, in the field, is very demanding to analyze or to collect roots in the field. It's not an easy task. And from the study of natural epigenetic spatial structure, we can say that it differs or can differ with the genetic structure, and indeed we, except for one case in which there is no relationship with the space, most of the cases, the slope of the relationships are different in these 42 populations. At intraspecific level, we can find that heterogeneous environments, as the one that we found for Erodium secretarium in Puerto del Tejo, promote epigenetic variation and makes the variability less easy to explain. At interspecific variation, we found that widespread species show higher genetic and higher correlation between genetic and epigenetic similarity. And this is also something interesting, meaning that probably the endemic plants that are in stressful environments have much more dynamic epigenetics that could be helping them to deal with this stressful environment. And yeah, just make the acknowledgement of the funding agencies, and okay, that's it. I think that was very fast, but more time for questions. You're welcome.
Thank you. Very interesting. I'm curious to know what you mean when you say that dispersal levels are similar. How do you know that? Is that based on syndrome classifications or?
Yeah, they are similar within the pair, not across the genus, okay? And that's why, so the pollinators, that will be one of the agents, are similar. So all of them are pollinated by insects, and we have conducted a study of pollination. We know the pollinators, and more or less they are similar. So not always is the main pollinator exactly the same, but in broad they are mainly bees and flies and little butterflies here. Okay, and then for the seed dispersal, either they don't have any, they share the system in the pair. So these, for instance, have fleshy fruits, and they can be dispersed by birds, and these have just seeds that fall down very close. So it's not the same in this direction, but it's the same within the pair. So the main difference between the widespread and the restricted is the habitat they live on. But they have a shared evolutionary history because they are in the same genus, and they also have similar pollinators and similar seed dispersal. That's what I was meaning. And thank you for the question, because I didn't explain it.
Okay, great. So you don't actually have data on seed dispersal. That's just basically assumptions based on population.
Yeah, we didn't follow the distance of dispersion, but the agents are similar between the pairs.
Looking for a parallel between phenotypic plasticity and ratio plasticity of these epigenetic alterations. That way, do you know, do you have got the role that these epigenetics can have local adaptation to the environment or the should have levels of adaptation or adaptation?
Yeah, that's very interesting. Indeed, epigenetics is like a mechanism behind plasticity, phenotypic plasticity. That's what I wanted to test with this experiment, because you can, in many cases, the experiments that have been conducted just look at methylation or changes in epigenetics, but without paying attention to the changes in phenotype or fitness. And then the evolutionary or the adaptive meaning of those changes in epigenetics are somehow unknown. Now we know that they can be adaptive or maladaptive. What we are, I didn't say, but we are following the two generations, so we knew the habitat of the previous generation, because the F1 also have two treatments. And now we are doing an experiment, a specific experiment, to have a higher power, statistical power, to detect this transgeneration transmission. And that's what we are conducting now in the greenhouse. It seems that in the most stressful population environment, the changes are adaptive. This is the population in which changes in the roots are higher, and in which the upper, above and below ground response were decoupled, and they managed to have higher fitness in the next generation subject to water stress again. But the other population have the opposite. Well, didn't reach a significant result, but it's non-adaptive. The changes, so if your mother has been stressed, then you deal worse in a controlled environment, and you are not more fit under stress. So the two populations show very no, because it's always like close to significant. But the power of the replication was not really strong. But not comparing species that are for, as we did, no? Some for stressful environments and the other not, and not always the response is adaptive. There is a transgeneration response, but sometimes it's maladaptive if you are not coming from a stressful environment, either if it's a population or a species. So yes, there is more tricky things to, it's a complex topic, and yeah, you have to be conscious of the enormous variability. So if you just sample individuals at different phenological stages, you can reach to whatever response. So we have always to sample the individuals at the same developmental states or the same age or these kind of things, because it's very dynamic. But yeah, not always well adapted, not always adapted, sometimes maladaptive.
So we have a question online.
Mm-hmm.
Ivan says, thanks Conchita for the seminar, very cool results. Do you know how big, different the genomes of the study plants are?
That's a good question. Between the species, we only know about the Erodium species. The others, in some cases, there are difference in genome size, but we don't know more than genome size for this. For these 6 pairs, we know the difference in genome size. I don't remember exactly now, but in Erodium in particular, we have the nightmare that the Erodium cathorlanum is polyploid. So in addition to have a change in genome size, we have polyploidy. What means that interpreting changes in alleles is more complicated because there are not just 2. For the others, we don't know about ploidy.
Any more questions?
Thank you so much, thank you so much, Conchita. Well, I was really interested by this like decoupling of the above ground and below ground methylation, and from a perspective of like life history and fitness, like longer term fitness and environmental effects on kind of demographic and fitness responses of a population. I wonder if you could speak to the fact that do you see that as a potential mechanism of how, you know, resource allocation might change in organisms in response to environmental change in a kind of lagged way with the environment? I don't know if you have, you know, looked at the kind of downstream effects of that decoupling on, as you were describing a little bit in this in your response to the question there, on the kind of fitness effects, but I don't know if you could speak a little more to that.
Indeed, root size, when we finish the experiment, we collect roots for fraction of the individuals, and root size was also changing. I don't know yet. No. Well, I thought that I could have the results there. Root size, the plants that have bigger roots were more fit above, okay? And the plants that are more dynamic and they count the couple above and below are the ones that are more fit also. So being able to somehow regulate independently seems to be useful. I don't know if adaptive, but useful in the greenhouse. In the field, I don't know.
Thank you so much.
Are there any more questions?
Yeah, thanks, Conchita, for the talk. I have a question about the experimental design, or how you sample in the first experiment that you commented. So you stress the plants, and you say that you water every week the control ones, and then every 2 weeks the other ones. So when did you sample? When did you collect the samples? I mean, because you think that the methylation pattern can have a dynamic and can change over the stress pressure.
I mentioned that we collect DNA and analyze DNA methylation at seedling states. At seedling states, the methylation was higher in leaves than roots. But at adult states, I show you that the roots were more methylated than the leaves. So it changed with the development, this.
But you think that, for example, in the ones that were water deprivated somehow, after each water exposure, they reset the methylation pattern, something like that?
In some cases could be, but the end of the experiment was when some of the individuals were already looking like senescence. So it was, all of them have produced flowers, fruits, and they are annual, so they are like finishing. They were not senescent, but they were reaching the stage in which you see that it's not longer to come, and then we kill all of them at the same time. But it was a mild draft. This year we have done a strong draft, and what we got was that 3/4 of the individuals died, and then we are now waiting for the survivors to be able to flower. But this it was they experienced draft for more than 1 month, twice, and they have suffered really a lot. But that's what we wanted to see this time, a real bad luck, bad season draft. That is, I mean, 4 weeks of draft is not something that is really astounding nowadays. But the first result is dying. The other didn't have that result. But and we have only used in this case seeds from Puerto del Tejo. So the one that is that we have seen is more suitable to do dynamic adjustment to increase the number of replicates. Because if you don't have a high replication, then the power of the statistical analysis is always in. So that's the thing.
Okay. Any more questions? If not, thank you so much for coming. Thank you again, Conchita, for the great seminar. And
Thank
see
you
again
all.
next week.
Okay, thank you.